In situ hybridization, or FISH, is a technique used to detect the location of specific genes or parts of genes on chromosomes within fixed cells. In this technique, a fluorescently labeled single-stranded DNA is used as a probe to bind parts of chromosomes that contain a complementary DNA sequence, referred to as the target. First, cells are arrested in interphase, or metaphase. The arrested cells are suspended in buffer solution and spread on a clean glass slide. Next, the target DNA and the labeled probes are denatured with heat or chemicals. This step separates the double-stranded DNA, leaving both the probe and the target DNA as single-stranded sequences that are capable of base pairing. Then, the probe and target DNA are mixed together and incubated overnight. This allows the probe to hybridize to its complementary sequence on the chromosomes. Once hybridization is complete, the excess probe is washed off. If the probe was labeled with a fluorophore, the locations of the hybridized DNA can be directly visualized with a fluorescent microscope. If not, the hybridized DNA needs to be fluorescently labeled before visualization. To do so, the probe sequence must contain a modified nucleotide bonded to a hapten, a small molecule to which the fluorophore, such as a fluorescent antibody, can attach. Such direct visualization of genes is very useful for cell-based diagnostic assays, where genetic abnormalities can be detected from any loss, gain, or rearrangement of chromosomes during cell division.
Performed. First, isolated tissues or cells are treated with a fixative, such as paraformaldehyde, to preserve chromosome morphology and integrity. Next, a chromosome spread is prepared, for example, by mechanically disrupting the material. The chromosomes are then dehydrated in a solution series of increasing ethanol concentrations and permeabilized in a pepsin solution to make target sequences more accessible to the probes. Chromosomes are then washed and stabilized with a postfix, denatured with formamide so that the now single-stranded DNA can bind to the probes, and dehydrated in a second series of increasingly concentrated ethanol solutions. Fluorescently labeled, highly specific DNA probes are prepared and mixed with unlabeled, repetitive DNA fragments to block non-specific binding. The probe is then applied to the chromosome spread, where it hybridizes to its target sequence, and unbound probe is removed with a series of washes. Finally, the chromosomes are mounted in a medium that preserves the sample and contains a general DNA counterstain, such as DAPI, that labels background genomic DNA, and a cover slip is applied. At this point, the samples may be observed under a fluorescence microscope, using the appropriate filter sets to visualize genomic DNA and sequence-specific features. After seeing how FISH is performed, let's look at some applications of this powerful technique. FISH may be used to confirm that an embryo's karyotype is normal prior to implantation. Here, a single cell, known as a blastomere, was biopsied from an embryo 3 days after fertilization, then lysed and fixed directly on a microscope slide. The chromosome spread was then hybridized to probes specifically selected to identify potential chromosomal disorders. In this case, deviations from the expected pattern of hybridization signals indicate disruptions in chromosome number or structure. Innovative techniques have also been developed to label all chromosomes from a single biological sample. One such method involves the octochrome device, an 8-celled glass slide preloaded with fluorescent probes. All 22 non-sex determining chromosomes, called autosomes, and the 2 sex chromosomes were labeled by distributing them among the 8 windows, each containing 3 chromosome-specific fluorophores. This allowed for concurrent screening of all chromosomes from a single hybridization. Finally, rather than separating the probes into different windows on a slide, all chromosomes can be labeled and then detected together using an approach called spectral karyotyping, or SCI. Multiple chromosome-specific probes with different fluorescent labels were hybridized to every chromosome, giving each one a unique spectral color. Concurrent observations of the combined chromosomes mixture are particularly useful for identifying karyotype defects, and may be used to identify aneuploidy, as well as chromosomal deletions and translocations. You've just